This is the nervous system part 3, what happens at the synapse. This is the third video for this unit. Again, refocusing on the idea that we are looking at this core concept. This is what we talked about in the previous video, the idea of the action potential and its different phases and what happens inside and outside of the cell, or more importantly, where do the ions move? And we should be able to kind of talk about this on our own at this point. Make sure that you can identify these, and if you can't, please bring this up in class. So what happens after this message moves down the axon? There's no such thing as, there is, but there are many neurons lined up to send a message typically. And so the question is, how does the message jump from neuron to neuron before it arrives at its destination, whatever that might be? We know that the signal is electrical, but the message sent between neurons is chemical. And the chemical synapse, which is the little space between the pre- and postsynaptic neuron, is basically a junction with a tiny gap that allows neurotransmitters, or chemical compounds that are a certain message, to move across that space. In most animals, transmission across that synapse involves these type of neurotransmitters: acetylcholine, epinephrine, norepinephrine, dopamine, serotonin, and GABA. This is kind of a close-up image of what your synapse is going to look like. The axon terminal is almost embedded in the dendrite of the next cell, but they do not touch. Basically, what we need to know is there are kind of 4 or 5 steps involved in this. The first is, once the electrical impulse reaches the end of this axon, the electrical impulse is basically going to send the calcium 2 vesicles towards the membrane at the end of that axon terminal. And we'll see that in number 1. And this is where kind of the change from electrical to chemical happens. During part 2, what happens is that vesicle carrying calcium is going to fuse to the, what I should say is the calcium then ushers the vesicles carrying the neurotransmitters to fuse to the end of the axon terminal. And basically they fuse and they're released into the synapse. The neurotransmitter is then in the synapse, and during part 3, during part 4, we see that the neurotransmitter is going to bind to whatever receptor is there, and it might open channels, it might cause another impulse to start, and then the neurotransmitter is going to be degraded, and it's going to be reabsorbed because neurotransmitters are actually reused. And this is kind of a close-up version of what that looks like. And this is kind of a verbal summary of that as well. Basically, there's an influx of calcium into the end of that cell, and that calcium causes the fusion of the neurotransmitter-filled vesicle to the end of that axon terminal. The vesicle releases the neurotransmitters into what we call the synaptic cleft, or that space, and then the neurons bind to the receptors, causing some sort of response. There are some really cool animated images that are similar to this on YouTube as well. This kind of looks at the last part, which is part 5. Typically, the neurotransmitter binding opens up the sodium channels to then depolarize the next cell, because we know that part of depolarizing a cell, part 2 and 3, and an action potential is allowing sodium in, and so the neurotransmitter allows for that to happen. Then what happens is the process of exocytosis, or the resetting. First, we have exocytosis, which means that the neurotransmitter is released, the synaptic vesicle fuses and releases the neurotransmitter into the synapse. Once they've done their job on the postsynaptic cell, they are degraded and they cannot be used during that time frame. What ends up actually happening is the neurotransmitters will be reabsorbed and reused. We won't talk about that much, but this is why we have a limited amount of certain neurotransmitters, and if we are lacking them, we may feel depressed or a variety of other things based on the neurotransmitter we're missing. There are 5 main groups of neurotransmitters that we need to know. The acetylcholines, the biogenic amines, amino acids, neuropeptides, and the gases. And a single neurotransmitter will have tons of different receptors. These are just the major known neurotransmitters that we recommend you know for the AP biology test. Take a minute, glance over this. I would definitely summarize acetylcholine, norepinephrine, dopamine, serotonin, GABA, maybe glutamate as well. I would know those. So to touch upon some of the most important ones, acetylcholine is really common in all organisms, vertebrates and invertebrates, and it is involved in muscle stimulation, memory formation, and learning. It is all around super important. There are 2 major classes of receptors that we see in vertebrates. So acetylcholine kind of has 2 major roles depending on which receptor it targets. Big picture for any neurotransmitter, our response is going to be either inhibitory or stimulatory. Stimulatory is going to push that response further. Inhibitory is going to stop something from happening. Make sure you take a look at this image and you are paying attention to what is going on and can label the different parts. This is an example question. Feel free to pause this and test yourself. So this is a little bit more of a in-close, more detailed version of what the cell body of a neuron looks like. And it takes into account the fact that there are both excitatory and inhibitory synapses, and depending on which one is put into action, that's what's going to happen. We know a neuron has a specific job, and I might want to make it continue to do that job, or I may want it to stop doing that job. And depending on what I want, I'm going to excite either my excitatory synapse or my inhibitory synapse.